Yeah. I'd like to introduce Professor Dave Towns. Dave and his team have been studying the recovery of islands following pest eradication for many years now, and particularly looking at the role seabirds play in that recovery. That's not just useful for those islands, it's also very useful for those of us that are involved in restoration projects. So, Dave.
Cool.
That should function. There's a...
Kia ora katoa. I'm going to sort of try to not be a data-rich presentation this afternoon. I think you're probably pretty much suffering from information overload. So I'm going to just talk about some general principles that we've kind of stumbled on as we've done this sort of work over the last decades or so. And then that will maybe serve as an introduction to the two videos, if we can get the videos to play, that have come from two students that have been working with us and with the University of Northern Illinois. So what I'm going to just talk about is sort of, given the time constraints we've got, only a few of the surprises that we've sort of stumbled on as we've done this work and learned about how island systems recover when you take introduced mammals out of them. So I'm going to sort of set the scene a little bit by talking about New Zealand islands as biodiversity hotspots, because you need to know how those faunas develop if you're going to do island restoration work. I'm then going to talk about the idea of drivers and passengers, how some things drive systems and other things sort of follow along in the wake. I'm going to talk about seabirds as social animals, so how they run their party lines. Some of you are probably old enough to know what a party line is. and also being neighbourly, how they affect other systems, and this can happen at lots of different levels. So I'm going to talk about a couple of those as well to finish off with. So there you go, that's how the thing is going to structure itself. You can do this in PowerPoint, that's why I did that. I'd like to thank, before I get started, some of the students that I've worked with. You're going to meet Lyndsay and another one, Christy Wails, shortly. Steph Burrell in the centre, and Rachel Buxton. I'll be talking about their work a little bit as well. And really what we've learned about in terms of how these island systems have developed has really come from a sequence of eradications of invasive species from islands in the Mercuries, starting in 1986 and finishing in 2014, plus a whole bunch of others around the coast. But these are the ones that have sort of been the focus of my attention since 1986. I'm not going to dwell on how important New Zealand is as a seabird place, because you know that already. You've heard plenty about that already today. But I do want to talk about a little bit the importance of place. Right? So where New Zealand sits has a lot to do with what you see in terms of diversity of seabirds. This is really neat sort of map that's come out of some work that Dennis Gordon and some others did a few years ago. And basically what it does is a couple of things. You can see this part that's in pink. That's part of the outline of the old continent Zealandia that James Cook actually came to find as the counterbalance for the Northern Hemisphere continents. He found it, but he stuck his anchor into it. Didn't realise that New Zealand is simply the high point of an old submerged continent. And the way things, the ocean currents operate around the current New Zealand archipelago involves pretty much southern cool currents in blue moving north and northern warm currents in red moving south. And I think Brendan showed a slide earlier today where there's a plume of chlorophyll that sits just off the coast in the Chatham Rise here. That's where those two currents converge. And when you get this nutrient-rich currents from the south meeting the warm, nutrient-poor currents from the north, they mix up, the nutrients bubble towards the surface, they reach sunlight, and suddenly magic happens. This is where the phytoplankton really get to work. And so around the coast of New Zealand, this is a hugely rich environment, not just for seabirds, but also for whales and dolphins. And things like all the apex predators that are associated with it, orca and great white whales. So That system has, or something like it, has been operating around what is now New Zealand for a very, very long time. The oldest seabird fossils in the world come from New Zealand. It's the old penguin fossils, 60 million years old. There's a little a paleo lake, which is now in what is it, central Otago, which was part of this bigger landmass 20 million years ago, called Lake Manuhirakia. A huge lake, and there's large fauna of native birds and also fish and various other things have been now found in the sediments of that lake, and included amongst those are fossils of what look like a diving petrel, and that's 20 million years old. So that system has been operating for a very long time. Included in that there's another couple of species that we'll be talking about in a few minutes, tuatara and native lizards, and they're also associated with seabirds. So this sort of arrangement is something which has a very long history. The other side of the coin is then looking at these islands from the terrestrial perspective is that we've got incredibly rich faunas on these offshore islands, far richer than you'd expect if they had been isolated for very long periods. The counterbalance to that is places like the Kermadec, where the fauna is naturally very, very depauperate, but have very high numbers of endemic species. So there's the loads of species of lizards, invertebrates, and of course the seabirds living on these offshore islands around New Zealand. And the reason we've got this rich fauna really is about the history of the island land masses themselves. And an example of how to view that is looking at where Te Aotearoa Toi, or Little Barrier, sits and has sat relative to the land mass of New Zealand in the past. So the edge of that green is the edge of the coast of the North Island of New Zealand during the height of the last ice age. It's not that long ago, geologically, 20,000 years ago. And as the ice melted and the sea levels rose, all these island archipelagos then started to get budded off. And so what you get ending up on these islands is basically a whole bunch of mainland species that got trapped. The end result of that is that you get quite big gene pools of variation squeezed up into a smaller and smaller space. So for that reason, we tend not to have much endemism on these offshore islands. The endemic species are the ones on the far outlying islands that have been stuck there for a long time and maybe started with very small populations to begin with. Then the end result of all of that, of course, is what we've got now is Little Barrier, 20 kilometres off the coast, seemingly hugely isolated from everything else, but with this history of actually being connected. So that gives you some background as to why we have these huge faunas of land snails, 13 species of lizards on Te Aotearoa Toi, and they can't swim. Tuatara are terrible in salt water, they hate it. So how did they get up to these places? Well, they didn't have to get out, they were there all along. So the first point I guess I want to make is that what you see on islands is this product of history. Now I'll talk a little bit about drivers and passengers. The way we've been talking about seabirds so far has been very much sort of on an ocean focus and what they bring to the land, and that's very much how these systems run. They run on imports. And what the seabirds import, of course, is the sorts of gut contents and things that people have been looking at. They don't consume anything while they're there. They're producers and not consumers in these sorts of environments, but they do have a huge effect on what the environments look like. So this is the summit, the highest part of Middle Island. In the Mercury group, the main culprit there is diving petrels, and they produce this landscape that's pretty much bare on the forest floor, riddled with burrows and a highly aerated soil, which makes it very difficult for many tree species to get a purchase. So you can see a milk tree lying on its side, sort of propped up by another one in the canopy. So the kind of environments that these sorts of birds produce are stressful for plants in many respects. There's super high concentrations of nutrients, very low soil pH, down to a soil pH of about 3, and very prone to wind throw. So the kind of plants that grow in these sorts of places have to withstand all of that. And milk tree is one of them. When they fall over, they'll just sprout from the base. And so you get relatively low plant diversity on islands with seabirds because you're dealing with these kinds of plants which some people might call extremophiles, things that can live in really tough environments. And they're extreme because they're also arid, very dry, which brings us to a bit of a paradox, because here's a seabird burrow service on Middle Island, and here are some of the inhabitants of those environments. Tuatara on the left, the robust skinks on the right. Both of those two species are actually very prone to desiccation. So how did they live in an environment which is incredibly stressful to them? Studies both on tuatara and robust skinks show that they have very high rates of water loss across the skin. Tuatara, when you hold them, feel like a piece of wet face cloth, cold and a face cloth that's been left and not washed very often either. And that's just the evaporation of the moisture across their skin. And they're not hot things to hold. The tuatara gets hot, it's dead. So how have we had tuatara in our fauna for a couple of 100 million years, and withstanding all these wild fluctuations we've had in climate over the time that the whole land mass has been present? And why are they living on islands with no water if they evaporate so much through the skin? Well, they live down seabird burrows. And if you stick a humidity probe down a seabird burrow, you'll discover that it's saturated for the whole time. So it's 100% saturation and a constant temperature of around 20 degrees through the summer. But if the seabird's in residence, of course, the temperature goes up a bit. So they've got central heating as well. Aircon and central heating, what more could you ask for? And it's the same really for these nocturnal species like the robust skinks. They come out at night when it's to be active, when it's damp, humid, dewy. Spend the rest of the time down the burrows where it's also damp. The top one up on the top left is the tusked weta, which is another one that uses these kinds of environments and the fact that they're heavily burrowed soils and extremely rich. There are the seabirds are the drivers, the tuatara and the lizards are passengers. But there are passengers also at a whole range of different levels. And this is another kind of passenger. These are plant parasites, scale insects. And you can see the threads from the larval scale insects on the right and central photograph there. Those are the anal threads. Those scale insects simply plug into the cambium of the plants, and the pressure of the sap moving up the plants provides them with their food. They don't even have to suck. So the food just goes into the guts, they digest it, and they extrude a droplet, and you can see one by that adult female in the middle of the program, photograph there. It's a droplet of honeydew on the end of that anal filament. So that honeydew is just packed carbohydrate. It's a huge energy source, and it's used by things like bellbirds, tui, and a whole bunch of different species of geckos. The tui and bellbirds that feed on the stuff during the daytime, because you can get the honeydew sort of out in the sun and the warmth, you can imagine what happens. It starts to cook a little bit, then it starts to ferment, and so you can get heavily drunk tui staggering around in the foliage of these plants after feeding on too much of the stuff. And you can then understand why tui gets so loud, right, and so aggressive. So they're both on a sugar and an alcohol high. So the interesting thing is, if you follow through the food chain on these islands, then here you've got a seabird modifying the soils, and you're ending up with a plant parasite becoming more abundant, because not only have the seabird modified the soils, they provided an environment where the host for these parasites predominate. So these plant parasites live on Karo and Nio. They jump from one to the other. This particular photograph was taken on Nio, but Karo has them as well. And you can quite often, if you go to these islands, see bellbirds defending these trees because they're so important as a source of food. Very easy to find out which trees have got honeydew scale on them because the trees are black. You will have seen them on Kānaka and Mānaka as well. There's that black sooty mould growth up the trees. That's simply mould benefiting from the sugars being produced by scale insects. So this is the kind of life cycle we've now sort of begun to understand as a result of these scale insects growing on two hosts, both of which are promoted by the presence of seabirds. And the interesting thing about this is Karo grows in very low light conditions, where Nio doesn't. So if you get an island where rats have taken out the Karo populations, which they do because they feed on the seed pods and plants simply fail to reproduce, you lose the honeydew scale, and so now we've taken rats off islands. We've got some islands with no honeydew scale at all because the Karo had disappeared. And so we've got, for example, someone mentioned Maui Mua or Lady Alice Island, that island has got loads of Nio, but no honeydew scale. And if you go to little rocks offshore that never received the rats, there you find Nio and Karo and honeydew scale. So this sort of complex web of relationships is something that we've sort of looked at in some detail for invertebrates. And on the right there, you can see that with the upward pointed triangles, the proportional difference between islands with seabirds compared with islands without. And the islands with seabirds have many times more of these 10 species orders of invertebrates that you can see on that figure on the left. And the interesting thing is that with seabirds, as I said, it filters right through the food web to parasites. Native wasps are all parasites of invertebrates, and they are much more abundant on the islands with seabirds, both in terms of numbers and species diversity, than they are on islands without seabirds. And the problem we have with island restoration work is that we can't replace those. When they disappear, if they drop out of the actual invertebrate fauna because of the presence of rats and the absence of seabirds, it's very difficult for us to figure out how to replace those native wasp species because there's so many of them. There's dozens and dozens of species of native wasps, many of which are partly flightless. So this is the kind of relationship between drivers and passengers that we sort of are starting to understand on these islands as a result of a lot of this work that's been done by taking introduced mammals off them. There are a large number of species that have shown lots of benefits just naturally. So species like morepork, kingfishers, and centipedes usually survive or can recolonise. But things like tuatara, of course, if they're knocked out of the system, there's no way they're going to get back. If we want that system to contain tuatara, we have to do it physically. So taking the next part then, and how these seabirds actually influence each other. What Rachel Buxton and Steph Burrell have found was that the old dogma we sort of started out with when I was working in the early mid-80s about seabird islands was that if you lose the seabird fauna from islands, you're basically screwed. The seabirds are so intensely philopatric that they will never recolonise. We've moved a long way since then. Some really nice DNA studies on grey-faced petrels, or oi, have shown there's actually a huge amount of mixing of populations of grey-faced petrels, and they move around a lot. And what Rachel and Steph have discovered is that you can actually get a lot of social attraction into these islands, and there's a lot more natural recruitment than you may think. And not only that, that social attraction isn't necessarily species specific. So as long as there's a party on, anybody who's flying by might just drop down to join in, regardless of if it's the same species or not. But that only works up to a point. And their main sort of what you might call recruitment halo for these sort of party activities is about 20 kilometres, 25 at the most. So then you can start to look at island groups where invasive mammals have been removed, and you can say, okay, if we know that, then we've got a fair idea of which islands in each group can get recolonisation from the neighbours. So here's one, a recruitment halo for the hen and chickens. It's pretty big. They overlap with the mokohi now, so there's likely to be quite a bit of interchange and high potential recruitment between those two groups. But the next big recruitment halo is further south. And what it does show is that Te Aotearoa, a little barrier, is right on the very edge of the range of natural recruitment. So that those other species that you might see on islands such as flesh-footed shearwaters may not actually be willing to go that far. The next big recruitment halo is around the aldermen and the mercuries. So in this part of the country though, the whole of the inner Gulf is well outside the range of those natural recruitment sources. So you'd have to think about, well, do we want to encourage birds into the inner Gulf? Some do it naturally, grey-faced petrels will, but the number of species that will do it is actually relatively limited by the look of things. So you can say we can rely on passive recolonisation for certain areas, and for others we may have to get more active about how it's done. If you don't do that, then you end up having to decide, well, what sort of environment do we want on these islands? Because what will happen is that without the drivers of that system, you will end up in what we call an alternative stable state. It's like it's a novel ecosystem which is seabird limited. And what that turns out to is something quite different from what most of the other island systems are like. Those novel ecosystems will actually have a higher diversity of native plants than the islands with the seabirds on them. It's kind of interesting. Sort of counterintuitive, because the seabirds actually are so dominant over how that ecosystem functions compared with these islands without them. So this is the effect of one set of islands on recruitment on others. And I just want to finish off with talking quickly about what we call transboundary effects. They're effects of one part of the system on neighbouring ecosystems. And they occur at lots of different levels. Here's this fine scale one, and Lyndsay's going to be talking a bit more about this in the video. But this dark shadow that you can see around Karapuki Island is actually a line of marine algae. And Lyndsay will be talking about what the effect is of seabird nesting on the islands on the marine algae adjacent to it. And it's a really interesting story. Steph has shown, and probably here's the burrow density on Karapuki Island, and probably the most useful one to look at is this one here, which is the concentration of nitrogen in the soils. So those darker areas are areas where the nitrogen concentrations are particularly high. And what happens is, of course, that you get runoff. Particularly during the winter, you get the islands basically flushed out by rain, and those areas of nitrogen-enriched soils filter down and end up in the ocean. And so they have an effect which is what we would say shifting from a terrestrial to a marine environment, and that effect can be tracked. And it's tracked using the same sort of stable isotope studies that people have talked about earlier today. So that's a very small scale, sort of a transboundary effect. And then, of course, there's the longer scale ones, which are the sorts of things that we saw in the earlier presentations. These are the effects of recruitment from New Zealand and then the birds moving into the whole Pacific Basin. We don't have many sooty shearwaters in the Mercuries, but we have other transboundary effects that, when you start looking at it, just this one island itself, as fluttering shearwater numbers, grey-faced petrels and flesh-footed shearwaters, and the others have increased as a result of taking the rats out, we're starting to have an effect over certainly the southern Pacific, and for some species right into the North Pacific as well, and across to the other side towards South America. So if you start to think about the fact that, okay, Karapuki is just 18 hectares, there's half a dozen other islands in the archipelago that are now also free of introduced mammals. They're all increasing in abundance on these islands. and then multiply that up to the number of islands from which introduced mammals have been removed from. Over 100. This is a seriously big global effect from what we're doing around the coast of New Zealand. And so the sorts of tracking work that's being done for all of these species is intriguing because it shows lots of things such as the potential for interaction with fisheries, the potential for species to interact with, for example, the Pacific gyre, the great rubbish patch, with all the plastic in it that can be affecting these. But when you start to do the maths, the biggest impact on any of these species seems to be the presence of rats and other introduced mammals on the islands. When you start to look at the actual effects on seabird populations, invasive species seem to be the biggest driver of any kind of threats that they have to them. And the biggest, for seabirds, the biggest combined threat is the combination of rats and cats on the same islands, because they feed off each other and then keep impacting seabirds. For some species, like some of the albatrosses, that may not be the case. There are other things happening which are affecting them, particularly inappropriate fisheries practices. But for most of the seabirds around New Zealand, it's certainly the introduced mammals. So there we are. What we've found is that island biodiversity is really influenced by currents and continents, where things are, where we are in space. We have these engineering effects of seabirds that hugely modify soils, but that leads then to a whole bunch of drivers that hitch onto the back of it. And by the way, with invasive species, the same thing happens. It turns out that you will get one particularly dominant invasive species, if you like, that changes the system, and a whole bunch of other invasive species will then hook in behind it because the possibility is there. And one intriguing example is the relationship between rats and wasps. Some of the islands that rats have been removed now have lost their wasp populations as well, and we don't know why. But, the next one is that the seabirds can recolonise islands. So this old idea of extreme phylopatry or natal fertility is probably no longer sort of one that we can hold. And finally, there's these multi-layered transboundary effects, both at small scales and at very large scales. And there's probably no other activity that we can do almost anywhere that has such a huge global impact for such a small area and such a relatively minor activity. So you people that are knocking over pests on headlands and all that sort of stuff are actually doing something that's got a global impact. And I think you should all go and talk to Bill Gates. He wants to make a global impact. Okay, that's my lot, and I'm going to use a terrestrial bird to say thank you. So we've got two students, both of who have been doing PhDs jointly between us at AUT and the University of Northern Illinois University in the States. Illinois, as you know, is is where Chicago is. You can't get further from the sea than Chicago. Right? But anyway, their best option is actually to come down here, because they've got plenty of sea down here, and they're using the fact that we've been removing pests from islands as a giant lab. So Christy will be the first one to speak. Yep. She's talking about her work in the Moka hen house, and then Lyndsay will be talking about her work underwater in the islands around the northeast coast. Okay.
I'm
Good
really
luck.
hoping this
Good
works.
luck.
It did load before while we were all having lunch, so I have my fingers crossed.
Hello, and thank you for being here for my talk. My name is Christy Wails. I'm a PhD candidate at Northern Illinois University in the US. Today I'll be sharing some of the work I have done as part of my PhD in this video presentation. If you have questions or would like to talk more about my work, please feel free to email me at wailscn@gmail.com. After the successful completion of island restoration programs, like the removal of pests from islands, seabirds will begin to establish breeding colonies. Interestingly, some seabird species are quick to arrive with very little, if any, human intervention to become established. However, other species require considerably more action from managers, ranging from the placement and maintenance of decoys to attract social species to translocating young to restored sites. Because restoration programs don't operate on an unlimited budget, understanding factors that influence seabirds' decisions on whether to nest at a restored island can help ensure best outcomes for our conservation dollar. So for my dissertation research, I'm examining how competition for key resources like food and nesting space influence the composition of nesting colonies on restored islands. Today, though, I'll be focusing just on nesting space. This work took place at the Mokohinau Islands, about 100 kilometers northeast of Auckland. This island chain consists of 4 larger islands with about 16 other smaller rocky islets. In the 1970s, some cattle and sheep were removed from the now retired pasture at Burgess, and in the mid-1990s, Kiore were removed across the Mokes. Since then, the seabird populations have been increasing, with 7 different species of pelagic tubenose species, as well as penguins that are now breeding at the islands. To understand seabirds' reliance on key factors in their nesting habitat, I worked to answer 3 questions with this study. How does habitat naturally change over time? How does seabird nesting activity, here measured as the number of burrows, change over time? And how does past human presence at Burgess influence habitat and seabird activity when compared to the other Mokes? So at Burgess, my collaborators and I conducted habitat surveys in 2009 and 2019 that involved identifying plants and estimating area covered by different plant species across transects and plots. And within those same transects and plots, we also surveyed for seabirds, searching for underground nesting burrows and identifying species that might be occupying them. Additionally, we conducted similar plant and seabird surveys at other islands in the Mokes in 2019. Today, I'll be showing the results for 3 of these islands. At Burgess, we are seeing a slow shift in plant communities. Over 10 years, we're seeing plant communities shift from being dominated by herby dicots to more shrubby communities. And over time, we're also seeing a slow but significant decline in the area that had been covered by invasive or non-native plants. When we look at seabird activity, here measured as the number and sizes of nesting burrows in study areas, we found a total percent increase of 62.5% in burrowing density across the entire island, with most study plots increasing from about 0.3 burrows per meter squared to 0.5 burrows per meter squared. The greatest increases were in small and medium-sized burrows, which would be occupied by diving petrels, storm petrels, and smaller shearwaters. Lastly, we compared results at Burgess to study areas surveyed on 3 other islands in the Mokes, revealing that the habitat elsewhere is quite different, largely dominated by shrubs and forests with significantly less grass and other invasive plants. Nesting density was, on average, lower than what we observed at Burgess, with a similar distribution, though, in burrow sizes. So returning to the original question, what factors influence seabird recovery? Habitat heterogeneity is key for supporting multiple species, and the presence of the large productive colony at Burgess is likely an attractive social cue to new dispersing individuals. So next steps for this project. I'll be adding in additional data from a recent trip to Fano, and also relating nest site selection at Burgess to chick growth rates and survival to fledging. Thank you all for your time. And again, if you have questions or would like to chat further, please feel free to email me at wailescn@gmail.com. Cheers.
My name is Lyndsay Rankin, and I'm going to present some research from my dissertation focusing on quantum influence in the nearshore environment of recovering seabird island.
Can everyone hear that?
I'm able to be there today, but if you
Not
have
so
any
good?
questions about my research, please feel free to email me.
Can we turn this up?
Successful mammalian predator eradications lead to the recovery of seabird populations. This in turn drives the entire system recovery through increased guano deposition. One way to track seabird influence on recovering islands is through stable isotope analysis. Now just some quick background information on nitrogen stable isotopes. The ratio of heavier 15N to lighter 14N can be used to trace marine versus terrestrial sources of nitrogen. Because marine food chains are longer, they have a higher ratio of 15 to 14N. You can use this ratio, known as delta 15N, to trace the relative amounts of terrestrial versus marine nitrogen in island systems. Systems with higher delta 15N have more seabird influence. This method has been used to study seabird influence in terrestrial systems. Previous research has found a positive relationship between delta 15N and year since eradication in soil, plants, and arthropods. As time since eradication increases, so does the seabird influence. However, there is little to no information on how seabird recovery affects the nearshore environment. We expect the nearshore environment to respond to eradication similarly to the terrestrial system. Specifically, we hypothesize that algal 15N enrichment as time since recovery increases. However, the marine environment is complex due to variables that may influence stable isotope measurements, such as season, depth, wave exposure, and runoff. Previous studies have documented the influence of guano in the marine environment at seabird islands varying in seabird density. Studies in the Baltic Sea and Hawaii have found high seabird mass density resulted in enriched algal delta 15N. Now, these studies can be used to predict nearshore response to eradication, since seabird density increases following mammal removal. To examine the influence of environmental variables on stable isotopes, we focus on 4 of the Mercury Islands, the islands varying in eradication and invasion history. The never-invaded islands have the highest burrow density, followed by the older and more recent eradicated islands. We established 7 sampling locations at each island treated. Sampling on Great Mercury was restricted to the south to avoid livestock influence, which is mostly in the north. At each of these sampling locations, we had a pair of scuba transects, and these were used to measure macroalgae species presence and cover within a 1 meter by 1 meter quadrat every meter in depth, up to 5 meters. This is repeated over the dry summer and the rainy winter. Algae samples from the 6 most common species were collected at each quadrat for stable isotope analysis. As expected, seabird influence was higher in the rainy season, the time of year when rain washes nutrient-rich guano into the near shore. Most species also had higher seabird influence closer to the shore, decreasing with depth due to mixing and dilution as samples are further from the island. 5 of the 6 species also decreased in delta 15N as wave exposure increased. More mixing from waves resulted in lower guano influence. And sites with higher runoff resulted in higher delta 15N, only in the red algae. Red and green algae have characteristics that allow them to take advantage of such nutrient pulses, such as high surface area volume ratio, high growth rates, and high uptake rates. The other study species were brown algae, which is typically slower growing and at lower uptake rates. Next, we increased the number of sample islands from 4 to 17 to determine the relationship between recovery age and nitrogen enrichment. Specimens were collected in the field, as well as sampled from the Auckland Museum of Herbarium. This is a great resource to quantify historical delta 15N in algae collected before and soon after eradications. A modified non-metric dimensional scaling plot shows how algal communities change with eradication time, here grouped in 5-year increments. And what this plot is showing is that each point represents a quadrat. Points on top of each other represent quadrats with the exact same species presence and cover, as opposed to points that are far apart from each other. Ellipses represent 95% confidence intervals around the groups. On the right-hand side in the green colors, the recent eradications had the least difference between the quadrats. And if you move counterclockwise, the older eradications and never-invaded islands have similarly high diversity and complexity between sites. As time since eradication increases, algal communities have become more similar to those at never-invaded islands. In terms of stable isotope analysis, we found 2 algal species that were influenced by eradication time. The samples were depleted in 15N as time since eradication increased, the opposite of what we were hypothesized and have seen in the terrestrial environment. However, Delta 15N values are still approaching never invaded values, even though they're lower. In summary, we found that multiple environmental factors influence stable isotope values in the nearshore environment, and that community structure and seabird influence through Delta 15N values approach those at never invaded islands. However, the direction is trending opposite to the terrestrial system. So more research is needed to examine the complexity of the nearshore environments. I would like to acknowledge everyone who helped with logistics as well as permissions and my funding sources. If you have any questions, send me an email. Thank you.
Thanks. I've got one of these too.
Oh, cool.
Edin's going to give the next presentation, and it's based on a report that she prepared for the Seabird Trust last year called Threats to Seabirds in Northern New Zealand. At the time it was released, it actually attracted an extraordinary amount of interest from the popular press, which gives us some sort of hope that when you put good information out, it gets well received. So thank you to Edin for firstly doing the report and for speaking about it today.
I'm not going to talk a lot about the actual threats review because it's here. There's a big stack of them down there, and you can all go and read it. It's got some really nice pictures in it too. I do want to touch briefly on it just to give you a reminder of why we did it, what it's about, what it contains, but what I want to talk more about is what is actually happening to address these threats, what we're doing, what was started before the threats review was reduced and what we're doing now to actually recognise that there's a lot of good work being done in our region to mitigate threats to seabirds. This has died, so I'm not going to be using that anymore. Okay. Backwards. This is what we have in the Aotearoa Gulf. There's the 27 species that breed here. Add 28, the Northern Pacific albatross or the Buller's albatross on the Three Kings for the whole of northern New Zealand. So this is what we have. This is what we have to lose, and this is what we have to look after. We've got an amazing diversity of seabirds here. We've got 5 species that breed nowhere else in the world. And we have the most endangered bird in New Zealand, the New Zealand fairy tern, that Chris touched on before. So the threats review, the reason we did this was there's nothing like this out there. There's nothing that condenses all of the threats to this particular group of seabird species in the world. It's available online as well if you don't want to have a physical copy of it. It is on the Northern New Zealand Seabird Trust website and the Auckland Council website. But the point of this was to outline the problems we're facing, to identify where we need to know more, not only about the seabirds but about the threats that they're facing as well. So we need this knowledge in order to protect them, and there are a lot of knowledge gaps which we've identified. Just to run through, these are the main threat groups that we identified in the review: invasive alien species, so predatory species, and island biosecurity, keeping our pest-free places pest-free, which is a real struggle. Fisheries, obviously a massive threat to a lot of our species. Pollution, within this we have light pollution, artificial light at night, plastics and potential for oil spills. Disease, climate change, and direct human impact. So this is disturbance both on land and at sea. And in the knowledge gaps we identified, obviously what we've been talking about a lot before. We don't have good ideas of how big these populations of seabirds are, how they're doing in terms of trends. There just hasn't been the long-term work, so we don't know what we're up against. We're also now obviously making good inroads into looking at their foraging ecology, their behaviour, and how we can manage them. But the other knowledge gaps that we have are around the threat prevalence and the severity. We don't know how much these threats impact particular species of seabirds. What impact does it have on, say, fairy terns? What impact does it have on black petrels? Because these are going to be really, really different based on the species biology. So what I'm going to do today is talk about what's being done to address the threats, breaking it down into the 3 sort of areas: research, monitoring, and restoration. These 3 bleed into each other a lot. You can't really do one without doing the others at the same time. But I've tried to break it up a little bit. We've heard a lot about research today. There's a lot of really cool stuff that people are doing in the Aotearoa Gulf, and it's a really good time to be a seabird scientist here. So we're looking at population sizes. Chris has talked about that. Biz has also talked about that, looking at how big these populations are, and then repeating these in the long term. This isn't data we can just go and get in 1 year and be like, there's this many seabirds. This is something that we have to revisit over a couple of years to get an idea of what the populations are actually doing in terms of breeding success. Some questions like that can't be answered quickly, but there are other things in terms of the impacts of threats that we can answer a little bit more quickly by doing experiments. One thing in particular that's going to be kicking off quite soon is the impact of different types of lights on nocturnal seabirds. So our petrels and shearwaters, we're near the biggest urban environment in New Zealand. There's a lot of light pollution, and we want to look at how different lights might be able to minimise the impacts on seabirds, both on land, so the urban environments, but also at sea. So massive cruise ships, there's one in the harbour today, and also fishing vessels. Monitoring is probably the most important of all these three things, because if we don't monitor, we don't know what's going on, and this is our problem now. We don't know what's going on. So we need these long-term studies repeating the same thing year on year to figure out what's happening with these populations. At the moment, we're establishing schemes for the long term. We're looking ahead 10, 15 years in the future. We can't just do this once and say, that's how it is. We've got to keep going back and doing it. And the methods that we're going to use are very different for different species. So a lot of us work on burrowing seabirds, on petrels and shearwaters. And the survey methods we use for them aren't going to work for gannets, like aerial surveys or white-fronted terns, species that are a lot more mobile and may breed in different areas in different years. So we need to develop methods to try and survey them over time. Restoration is monitoring. The monitoring side of restoration is how we know what we're doing is working. So for example, at Tawharanui, we've had some long-term monitoring now of grey-faced petrels, doing mark recapture, seeing how that population is growing since the predator-proof fence has been put up there. And we have had setbacks, but we do have a graph that goes like this of the breeding of seabirds there. And we've got grey-faced petrels, common diving petrels, and these extraordinarily cute fluttering shearwaters. I am a little bit biased, but they are really adorable when they're small. And it's really exciting to have these birds back on mainland New Zealand. So the monitoring we're doing there is encouraging us that we're doing the right thing, and it's hard work, and it does involve constant vigilance because there are incursions and we need to prevent more predators getting back into this area, but it's going well. So restoration, this is the active work, this is the grunt work that everyone's doing that involves, it's not all about nest boxes, everyone seems to latch onto nest boxes and they are really useful, but this is also the long-term predator trapping and restoring coastal vegetation so that these areas are good for seabirds to come back to. There's no point in trying to attract seabirds back to an area if we haven't mitigated the threats to them that drove them away in the first place. And it's a huge effort. I was down in Raglan recently with Carrier Project and the Burrow Buddies who are doing an amazing, amazing job trapping to try and keep these birds, grey-faced petrels, breeding on the mainland, and it's just constant. I was there 2 years ago and we scoped a lot of burrows and found the first chicks there, and I will never forget the look on Crystal's face when we found the first grey-faced petrel chick in a burrow down there. This year wasn't so good. We went back and we scoped a lot of empty burrows because they'd had a ferret come through and luckily picked up on it really quickly and caught it really quickly. Unfortunately not fast enough to save some of the grey-faced petrel chicks there. The thing is, pest problems don't go away when you put up a predator-proof fence. Even if you do a lot of trapping, you're still going to have events like these, but it's not the end of the world. A lot of these birds are really long-lived. Grey-faced petrels can live for 40 years. They can breed into their 30s, and they're going to keep trying. So even if we have one bad season, it's not the end. We've just got to keep pushing to look after these birds. I'm highlighting a few different threats here because they're going to be really different for different species. Like I mentioned before, threats to burrowing seabirds on our pest-free islands are going to be really different. They're going to be more at sea based than the ones that nest on land. Things that I think are particularly important in our region in Auckland, there's a lot of people here. There's a lot of human disturbance to birds on the mainland, like fairy terns nesting north of Auckland on beaches, Pakari Beach, Mangawhai. So they're really impacted by that. Also, a lot of our seabirds are impacted by boats out on the water in the Hauraki Gulf. There's a massive recreational boat use here, and that is actually negatively impacting some of our seabirds. The other thing is lights, because this is an area of pollution that sort of gets ignored. Everyone's really interested in the impacts of plastics on seabirds, but lights are a real problem for seabirds at night, nocturnal seabirds like petrels and shearwaters. Last year we had an incident where 76 shearwaters landed on a cruise ship vessel and over half of them died because they weren't handled properly. Bird Rescue did a really good job of looking after the others and releasing them successfully, but as a response to that, DOC came up with this seabirds landing on ships guides that's been put forward to the cruise industry or the crews of the ships who will have to deal with incidents like these. And we hope that by pushing to get this knowledge out there, to teach people how to properly deal with incidents and handle seabirds and look after them, that we can reduce the impact of incidents like these on our seabirds. I'm talking a lot about other impacts on seabirds, and the reason we're doing that is because at the moment the Department of Conservation and MPI are currently drafting a national plan of action related entirely to the threat of fisheries on seabirds. And this is a huge threat to a lot of our most endangered species, but it's not the only one, which is why this is not just about fisheries, it's about all the threats that our seabirds are facing, both at sea and on land. I do encourage you to read through this and make a submission. It's currently open to the public so you can see what is planned. I wanted to leave you with something positive and adorable, because researching threats to seabirds and working in conservation is often really, really depressing. And there's a lot to do. There's some real mountains to climb, but we are making steps, and now's a really exciting time because people are really keen to help. If you don't feel like reading quite a long and dense report, then next year we have commissioned this wonderful person, Giselle Clarkson. She's an illustrator. She's an absolute extraordinary communicator of science. She's a generally excellent person. I know she's one of my best friends. But we've commissioned her to do a comic treatment of the Seabird Report, which will hopefully be out sometime next year. So look out for that. Cheers, everyone. This thing's dead, so I'm just going to get rid of it.
Okay, cool.